Hello, good afternoon. Good afternoon and welcome to the second annual High Point Clinical Trial Center webinar focused on advancements in NASH. My name is Lorraine Rush and I'm president of HPCTC. We're really excited to host the second webinar, which is deeply aligned to our metabolic focus and early clinical research at our phase 1/2 facility in North Carolina. I will keep my comments brief and we'll move right into the webinar at hand. As most of us know, NASH is a medical condition predicated on the increasing buildup of fat and inflammation in the liver and is closely related to obesity and diabetes. Largely undiagnosed, it borders on an epidemic and is currently a top target of drug developers globally. In fact, it was a clear-cut highlight at the recent JP Morgan investment conference last week with early data released from multiple companies. There are multiple challenges in this therapeutic area, and today's goal is to share current thinking in the specific areas of clinical research. Specifically, we'll look at the best regulatory approaches for soluble biomarkers, as I call it, to validate or not to validate, emerging platforms for patient screening and clinical patient enrollment and recruitment strategies to successfully enroll patients for early proof of concept studies. On a note of housekeeping, the format for today will be 3 presentations of approximately 15 minutes each, followed by 5 minutes of questions. The attendees can share their questions via the chat window. Time permitting, we'll take them directly after the presentation for approximately 5 minutes. After the presentation, all of the presentations complete. Additional questions may be addressed at the end of the webinar. And if your question is not addressed due to time, no worries. We will actually follow up with a Q&A document circulated post-webinar, and the recording will be available as well. So on to our first speaker. Speaker 1 is Dr. Anika Carlson. We're really excited to introduce her. She's from Mercodia, and her expertise is in the spectrum of ligand-binding biomarker qualification and validation as the head of bioanalytical services. Combined with her research in beta cell damage pathways, she's an ideal speaker to share strategies around the development of appropriately regulated biomarkers for NASH. And Dr. Carlson, I'd like to turn it over to you.
Hello everyone and welcome to this case study on how to improve measurement quality, throughput. Hello everyone, welcome to this case study on how to improve measurement quality, throughput and cost effectivity. So the outline of my talk will be a short introduction on the biomarkers in NASH. I will speak a little bit about the guidelines and regulations in bioanalysis and touch upon validation, qualification and fit-for-purpose validation, and how to start building the bioanalytical strategy. And then we will move more specifically into a case study of Glucagon as a biomarker in NASH, challenges and mitigation, and a summary. So there is an unmet need to develop biomarkers that facilitate research in different aspects of NASH, including the diagnosis, identification of populations at risk, assessments of disease progression or regression, and also response to treatment. And this has been excellently summarized by Sanyal et al. In Journal of Hepatology. And Sanyal also summarizes that there are numerous stakeholders involved in developing strategies for NASH, including both academia, the NIH, pharmaceutical and device manufacturers, such as our company, developers of diagnosis, the FDA, healthcare providers, professional associations, but also insurance providers and the patients. So, how can a bioanalytical service partner assist in biomarker assay development and validation? Well, we believe we can help by developing a bioanalytical strategy together with the sponsor. And the first we would do is to try to understand the intended use of the biomarker data, because the intended use should guide the choice of method development and validation strategies. And there are quite many different guidances and regulations in bioanalysis. We have the guidelines on bioanalytical method validation and sample analysis that's handed out by the FDA and the EMA. For the preclinical field of GLP, we have the OECD directives. And on the clinical side, we have the WHO guidance and the CLIA. And for the manufacturers, for medical device, for example, we have the ISO. So there are many different guidances that adhere to, and they are slightly different, but they're also similar in many ways. And some of them, for example, the EMA and FDA, they speak about the validation. And the word validation, that can mean many different things. For example, in the context of drug manufacture, it's documenting that a process or system meets the predetermined specifications and quality attributes. But in a bioanalytical context, it usually means the fulfillment of predefined acceptance criteria, for example, curve fit, accuracy and precision, selectivity, stability. And this is something that you can find in the FDA and EMA guidelines. Sometimes you also see the word qualification in the context of biomarkers. And qualification, that can also mean many different things. For example, if you speak about clinical drug development, the context of using a qualified biomarker could be the demonstration that a biomarker reliably supports a specific clinical interpretation or clinical application. But it means a very different thing in the bioanalytical context, because in the bioanalytical context, we usually speak about a limited set of validation experiments and acceptance criteria. And these validation experiments are selected based on the intended use of the data. And this is actually the same thing as when we speak about fit-for-purpose validation, because in a bioanalytical context, the fit-for-purpose validation means a limited set of validation experiments selected based on the intended use of data. And this has been excellently summarized by Lee et al. In Pharmaceutical Research from 2006, and more recently discussed by Safavi on the Bioanalysis Zone website. So, how can we understand the bioanalytical guidelines and interpret them? Well, they both, the EMA and FDA, outline development validation strategies that are accepted for regulatory submission. And a full validation is expected for studies submitted in new drug applications that require regulatory decision-making for approval, safety, or labeling. But actually, the guidelines also discuss fit-for-purpose validation that may be used for more exploratory methods that don't support regulatory decision-making. And a couple of examples here would be candidate selection or internal decision-making. But the sponsor also needs to consider for a specific study that might be validation that needs to perform that could be scientifically relevant, for example, specificity or stability, that might not be necessary from a regulatory standpoint, but very important for the design of the study. So, how can you start building the bioanalytical strategy? Well, we would suggest to initiate a partnership with the bioanalysis lab as early as possible and exchange as much information as possible. So to start with, the intended use, and describe the study. Is it for primary endpoint decision, regulatory decision for approval, safety, or labeling? If we are in the preclinical field, is it GLP or non-GLP? Or is it rather discovery exploratory? And what is the desired specificity? Are there any interfering substances, concomitant medications? And what is known about sample collection, handling, stability, and storage? Do you have a preferred choice of assay or platform? And least but not last, timelines, because the timing of development and validation of methods in relation to the study protocol and sampling dates will be very important. So, moving into a specific case study of an interesting biomarker, I will talk a little bit about glucagon in the search for new biomarkers in NALFD NASH. So glucagon is mostly known as a counter hormone to insulin and to be involved in the regulation of glucose homeostasis. However, in a couple of recent publications, it has been shown, for example, by Soloway et al., that glucagon may have a very interesting role in hepatic amino acid turnover and serum amino acid levels. So Soloway have shown that amino acids may stimulate glucagon secretion and actually promote alpha cell proliferation, and that glucagon may act on the liver. And in another publication by Junker et al., it was shown that increased fasting glucagon levels could be seen in AFLD in NASH patients independently of diabetes. So this has been followed up by other publications from Jens-Juel Holst and Weaver Albertson on kind of a feedback loop mechanism where glucagon acts on the liver and the amino acids acts on the pancreatic alpha cells. So a hepatic glucagon resistance has been associated with an increase in liver lipid infiltration. And numerous studies have pointed out altered levels of plasma amino acids in NALFD. Hepatic fat accumulation and insulin resistance leads to an impaired effect of glucagon on plasma amino acid levels. This increased amino acid concentrations give rise to increased glucagon levels and hepatic glucagon resistance. And the elevated fasting glucagon level in humans is suggested to actually indicate a non-optimal function status of the liver. And the glucagon-alanine index has been proposed for assessing impaired hepatic glucagon sensitivity due to impaired liver function. So if you want to measure glucagon as a biomarker, what do you need to know? Well, glucagon is expressed from a very complex gene coding for many different proteins, including not only glucagon, but also GLP-1 and GLP-2, proglucagon 1-61, glycentrin, and oxyntomodulin. By an intrinsic post-translational processing in different tissues, all these different proteins are generated from one single gene. And actually a number of proteins contain the full sequence of glucagon. And this makes it, of course, tricky to measure glucagon specifically. And it's also very conserved among all mammals, and it has issues with stability. So, the challenges and mitigations with glucagon. Well, first, you need to consider the choice of assay and platform. What kind of specificity do you need, throughput? What is your sample matrix, and what performance do you need? And we addressed a couple of these questions in the Mercadia glucagon ELISA by using mouse monoclonal antibodies with excellent specificity profile and low cross-reactivity with proteins with a similar sequence. And we also optimized the assay protocol for best throughput. And we have sample stability data available in different matrices. And also assay performance data is available. And this is really an advice when looking for commercial assays. What can the manufacturer show in terms of performance data? And then you need to, of course, consider your validation strategy. Is it exploratory work, or is it something that will be built on regulatory decision? And for example, validating an many different biomarker assays. You have challenges, for example, with the calibrator matrix, because you usually have the endogenous proteins available. There are not any donors that don't have glucagon in their blood. So in many cases, you need to use surrogate matrix, and you need to show data that you have excellent performance, such as parallelism between your calibrators and your samples. And we have addressed and performed the full validation according to FDA and EMA. And these validation results are available for decision making or validation on site. And something that's also discussed, especially when it comes to ELISAs, that is the life cycle management of assay reagents. For long studies over several years, you need to secure that you have a low lot-to-lot variation and that the manufacturer you work with can ensure reagent control. So, in summary, measurement quality, throughput, and cost effectivity can be improved by forming bioanalytical strategies, and also by collaboration between different stakeholders involved in searching and validating new biomarkers in NASH. Thank you.
Hi, thank you very much, Annika. I wanted to follow up to see if we have a little bit of time. There are no questions out there for Annika at the moment, but I'll pose one myself. I guess the question I had for you is, in the amount of work that you've seen over, just say, the past 2 years, Doctor, what do you think the percentages of fully validated versus qualified endpoint assays that you're seeing as part of development programs for your clients?
We have been working mostly with clients that really want to send IND applications, for example. So it's been important for those clients to have fully validated assays. And I would say during the years I've been in the field, there has been an increasing interest also for fully validated biomarker assays. And I think it will be a further increase due to the discussion and the different bioanalytical meetings, and also the new FDA guidance that speaks more about biomarker validation.
And do you have any comment on, I mean, many of the exploratory data points that we're putting in there are really safety labs, right? So where does a safety lab end and an exploratory biomarker begin? And, you know, is there a thought process for how you would apply more than just the fit-for-purpose qualification to some of those, or is that really on a case-by-case basis? That's one question. And the other question I had is, maybe especially for people out there that are trying to understand for their programs, what the difference in period of time that it takes to qualify in a fit-for-purpose manner a commercially available assay versus even, you know, validate that commercial assay or revalidate and build a new ligand binding method. I think that a lot of people and need, you know, would be interested in, for planning purposes, the length of time and commitment that takes.
Yeah, and usually the as I mentioned in my talk, you need to contact the bioanalytical lab early to address these questions. And for the first one, I will say it's of course a case-by-case decision on how to validate. So I would say that the first thing is to really discuss, and also the intended use of the data. It could be absolutely good enough with fit-for-purpose validation or qualification, if that's enough to support your data. And you shouldn't overdo, because that will only cost you and in the end also cost the patients a lot of money. So it's very important to choose the appropriate level and don't do more than you have to, because that would be just a waste of money, and do the full validation when it's really needed. And there, of course, there's very different timelines for developing an assay from scratch, or do a full validation or a fit-for-purpose validation. There are some differences also between, now we're speaking about ELISA, there are some other methodologies using in bioanalytical labs that might be faster. But method development at least is several months, and a full validation is something around, 2 months or something. You should at least have contacted your bioanalytical laboratory, and less than that if you do some kind of qualification. That's very general, of course.
All right, I think that's a good overview, because, as we're joining drug development teams and we're on, the drug development program, the question is how long, from the project managers, how long is it going to take to validate your ligand binding assay if you go down that route? And so, it does take time. So 2 months for the development and validation as a minimum, right? Especially depending on the resources.
For development, I would say something around maybe 6 to 8 months, even though if you start with a new development.
Right. So depending on how far you are with the current method that you're using, and there's a difference between transferring an internal method to, and then developing or optimizing that method and validating it. And some of the internal methods do not transfer well or aren't completely developed when they're transferred, so that takes additional time. So if you're really looking at starting from scratch, you're looking at a 6-month lead time, correct?
Yeah, at least. Yeah, that will depend a lot on what kind of reagents you have available, of course.
All right. Well, I think that addresses the question. And thank you very much, Dr. Carlson.
Thank you.
I'm going to move over to our second speaker, Dr. Remco Enserich. He is the U.S. Director of Medical Affairs for EchoSense. As an epidemiologist and a statistician, he's focused on the application of non-invasive screening and diagnostic tools such as FibroScan and the support of patient selection into NAFL and NASH clinical trials. And he'll be sharing those strategies with us today. And I'm going to turn that over to Dr. Enserich now. There it is. Thank you.
Great, thank you, Lorraine, and thank you to the organizers for letting me speak today about the FibroScan and specifically working principles, utility, and also collaboration in the NASH clinical development space. I'll talk a little bit about the rationale for using FibroScan in NAFLD-NASH clinical trial setting before anything. I think to many people, on the call, it's pretty clear that NASH today is a highly competitive space. And I borrowed this graph from Monica Korneman from Journal of Hepatology in 2018. That basically shows that there are a lot of different pharma players in the market, but while the mechanism of actions for different pharma companies may differ, most players do look to show some form of fibrosis improvement down the line. And the reason for that is pretty clear, has been shown by Late Angelo in Gastroenterology in 2015, already very good paper. That basically shows that liver fibrosis, but no really other histological features so far associate with long-term outcomes of patients with NAFLD. And so when you, for instance, look at patients with NASH F1 compared to patients with fibrosis and F4 and NASH, the risk of dying or transplant is about 5 times higher. So this is basically the reason why so many players are focused on fibrosis improvement to some extent. Now, there are many different ways that you can assess fibrosis, obviously, and I'm not showing all of them here. For instance, I'm not showing any of the blood-based markers available. But before the 50s, obviously, we only had percussion, which is obviously a pretty insensitive technique. After the 50s, and still today, we rely heavily on liver biopsy, which is the golden reference in many of the pharma trials out there, but has pretty high cost, high risk sampling error, and is definitely not very suited for follow-up longer term once drugs hit the market. So after 2002, multiple elastography techniques came to market, including the FibroScan, and great sort of like added feature, augmenting potential liver biopsy later on. Very easy and fast, non-invasive in real time with a relatively large sample size. don't have to be an ultrasound tech when it comes to the FibroScan technology at least, and very suitable for screening and follow-up. Now, obviously, also not everything is perfect. Elastography and also FibroScan to some extent is subject to confounding factors such as inflammation, cholestasis, non-fasting, etc. And it can only be used to stage basically the fibrosis extent and not so much the fibrosis architecture, which might be important in some cases. So, that's sort of like the rationale for FibroScan and NAFLD-NASH trials today. So I'll speak a little bit about the company and the product timeline and then the working principle. EchoSense has been very successful since the generation of the FibroScan. Today, we are a global company with over 250 employees that is active direct in 8 countries. We have over 4,800 systems installed. We recently hit our 1,000th system mark in the US. We are headquartered in France with more than 120 people there. That's where a lot of the R&D and manufacturing happens, but we also have a pretty strong presence in the US, in the Waltham office, which is actually where I'm based too, mostly sales, clinical, and service, as well as in China. In terms of product line, we have the birth of VCT, or vibration-controlled transient eagle elastography, which is basically what dictates the FibroScan stiffness measurement today, was in 2003. And since then, we've come a pretty long way with the generation of additional probes to basically be able to scan in a variety of patient populations, including NAFLD with the XL probe. We generated the CAP, a module, which is basically the controlled attenuation parameter that I'll speak a little about further later on. And more recently, you can see that we are getting more and more focused on smaller battery-powered systems, such as the 530 Compact and more recently the 430 Mini. So we're moving to smaller and smaller systems. All these systems, basically, whichever FibroScan you would be looking at, model or software, allows you to non-invasively quantify 2 physical biomarkers within roughly a 5-minute exam, assuming that you know what you're doing. And basically, you take the probe that belongs to that patient, whether that's an S for a pediatric patient, an M for a regular adult patient, or an XL for an oversized patient. You put that on the right side of the patient, you hit the button, and that basically induces a mechanical shear wave that travels through the liver at a certain depth below the skin. And basically the speed with which that wave travels is measured by ultrasound, which is also embedded within the probe. And that then gives you a readout of 2 things. First and most important, a correlate of fibrosis, the liver stiffness measurement. And secondly, a correlate of fat or steatosis in the liver by the controlled attenuation parameter. And these 2 parameters are then saved in a report and can be used by the clinician in conjunction with many other biomarkers to assess the liver disease severity in clinical trials or otherwise. So that's basically a little bit of a background in terms of how FibroScan is used in NASH trials today, whether that is early clinical development, phase 1, 2, 3. The idea is mostly the same in all of these trials when it comes to the FibroScan, and that is that FibroScan in all these situations basically allows for very efficient pre-screening of patients into NASH trials. So based on internal data, we know that if you use a FibroScan for pre-screening before the actual screening into a protocol of a pharma trial, that the volume of patients that you can push through is about 30% higher, but also the success rate in terms of the patient randomization into the trial is about 40% higher. And the way that companies that do pre-screening with FibroScan work is basically they take a population of suspected patients with NAFLD. They scan them with a FibroScan in a pre-screening situation, and then patients are basically put on an axis from low to high CAP. That was one correlate of the FibroScan, and high stiffness, low stiffness. And basically then the pharma company, depending on the patient population of interest, will select based on those markers which patient is suspected of NAFLD F0, which patient is suspected to be healthy, or NASH F1, NASH F2 or 3, etc., and NASH F4. And that then, together with additional testing, including of obviously a liver biopsy, will lead to patient inclusion into the trial. So this is just a little different way of looking at that. The practice can help the sponsor very much in finding the right patients in such a situation. So the idea would be that you don't want to push every patient or the practice through fully consenting a potential patient of interest if you don't know anything yet about the disease status. So you do an abbreviated consent that basically says, okay, we're going to do a fiber scan on you, and if that fiber scan sort of like has certain values, we might consider full patient consent. And so that basically stratifies people into no participation, meaning not even eligible for full patient consent, or into full patient consent, and then basically clinical testing, the liver biopsy, and then the screen-in and screen-out situation. So that is basically how FibroScan is used in most phase 3 trials today, but also in earlier phase 2A trials and 2B trials. From a site perspective level, the way that these protocols often work, because now we're looking after full consent within the study itself, there might be a FibroScan to reconfirm basically the pre-screening. Again, then there will be a second screening, obviously, with the liver biopsy, blood draw, and then office visit. And throughout the protocol of the trial, you'll have multiple office visits, then a closeout FibroScan, biopsy, blood draw, etc., and you're done. So this is just one example that we've seen. This is for a phase 3 trial. And the only reason that I don't show any early other examples in earlier clinical developments because I don't have them. But it does show that FibroScan has a minor role to play in the revenue generation of any site that is involved in pre-screening or follow-up of patients during the trial, although the numbers may vary greatly depending on the contract with the pharma company. So finally, a little bit about pharma research collaborations that Echosense has with many of the pharma players. As you might know, FibroScan is a very strong clinical legacy that is supported by over 2,000 publications today, and also mentioned in many of the key guidelines, including AASLD, WHO, and AGA today. And so what we often do with the pharma companies have a very open discussion on the front end together with our biostatisticians and our medical affairs teams to understand for that pharma company, for that protocol, what is the population that you're working with, how do those patients flow into the protocol, what are the metrics of interest that the pharma company might be looking for, and then based on internal databases that we have, because of all those publications that we have in the field today, we help the pharma company to generate very advanced and tailored scoring systems that basically suits their needs. So more recently, in 2018, a couple of those were presented at AASLD as a result of those collaborations. And they basically speak to, triazing scores for NASH. So a lot of players are interested in the NASH F2 patients. So we developed algorithms for that. It might be also to determine basically reducing of screening costs, etc., into the trial. It can be around screening, as it most often is, but it can also be about monitoring performance. So basically, we work in a very open way with the different pharma companies, depending on what they need. And so now moving a little bit to AASLD, which obviously we recently had in November, and a couple of those collaboration activities were generated into an oral and presentation. So one of the things that we generated was the FAST score, which is basically a combination score of the liver stiffness measurement of FibroScan, the CAP measurement of FibroScan, and AST. So here you have a score that is a combination of fibrosis, inflammation, and steatosis. And based on internal data sets, but also on external validation cohorts, it was shown that this particular FAST score is very well capable of identifying patients with NASH, meaning a NASH score of greater or equal than 4 and advanced fibrosis F2 or higher. Didn't really matter which country we were looking at, and not even the patient characteristics, even though they may have differed greatly between the bariatric surgery cohort, for instance, as one of the external validation cohorts, or a Malaysian cohort or US cohort. Cohorts were consistently good or excellent in most of these situations. So that was a very interesting, and that was an oral that was presented at AASLD. Another example, also again based on collaborations with pharma, was that we realized that a lot of pharma companies are interested in basically reducing the screen failure rate while keeping the missed case rate as minimal as possible. And so this is an analysis again that we did on internal data sets with the pharma company together to say, okay, compared to no strategy at all, if we decrease the screen failure rate by increasing basically the cutoffs of stiffness, AST, and CAP, what level of screen failure versus missed case rate are you comfortable with? Which level of total costs that you need to expend to find that population of patients are you comfortable with? So this was very helpful for the pharma company to determine where they wanted to be in terms of enrollment time and cost decrease. Okay, so that was also an ASD presentation. So then finally, I'd like to close out with just an analysis that we did on the AASLD booklet, where we tried to figure out presentations on our technology and other competitor technologies. And it's pretty clear that Equisense has a very strong role to play in the elastography space. This is the number of abstracts that we found in the AASLD abstract booklet, roughly 150. So great deal of buzz around FibroScan technology. When you break that 150 roughly abstracts down, you can see that most of the abstracts focus on HCV. A little surprisingly, maybe not so 2 years ago, but there's still a lot of focus on HCV today with FibroScan. That's because FibroScan is used as a reference technology basically these days for fibrosis. But we see that NAFLD NASH related papers or abstracts are increasing, and especially this year was a very good year in that sense. So last slide, basically to close out, is the fact that we have a lot of ongoing pharma-centric collaborations, and one of the things that we realize is that within pharma, it's not only about screening of patients into trials, it's also about monitoring patients over time. And so FibroScans might have a role to play in that scenario. And so what we also know is that the drug effectivity on fibrosis or NASH regression is probably going to be subtle by itself. And so any reduction in FAST score or stiffness or CAP will probably also be subtle. And so if you want to pick up on such a signal, you need very highly standardized and efficient training products, because you don't want to get it right once during screening. You need to get it right maybe 10 times throughout the study period. So as a result of that, Equisense decided to build a dedicated team of clinical specialists that we have available to us in the US today, and that basically, if requested by pharma, can come over and give FibroScan demo meetings, FibroScan demos and investigating meetings, national sales meetings, study coordinating meetings for pharma companies. So there's definitely a lot of interest from Equisense to collaborate within pharma. So I'll leave it at that. Thank you.
All right, thank you very much, Dr. Ansrig. There's FibroScan is one of the emerging technologies in the space, and the question is, how does this fit between research development and endpoints? We have a couple of questions from the panelists. One of them, and I think it relates to your last point very well, is does Equisense view FibroScan as an eventual replacement for biopsy, or how do you view that the future development of FibroScan in relation to drug development?
Right. So I think what we're realizing over the last couple of years is that no one biomarker will be able to replace biopsy per se. I think if anything, FibroScan will be part of a larger algorithm or score to identify patients. So that would be one. It's not about FibroScan per se alone. The second thing is that I think from a screening perspective, I'm not so sure that we will ever replace liver biopsy, because as I mentioned, we only measure liver stiffness, and while liver stiffness is sort of like a surrogate of fibrosis, it's not fibrosis per se, and we also don't really give any information around the nature of the fibrosis development. So we only see collagen content basically, not so much collagen architecture. So I don't think we'll replace it in that sense. When you look at a more prognostic outlook, using FibroScan as prognostication, I do think there's a role to play for FibroScan there. There are very strong articles out there these days that show that even within the cirrhotic range of kPa values per FibroScan, there's a great deal of variation in risk to mortality, for instance. So if you're a 12 kPa versus a 50 kPa versus a 75 kPa, it's pretty clear that the mortality risk of these patients actually differ, and that is something that might not be able to be picked up by a biopsy, which only has an F4 category in that sense.
Right, and that's one of the challenges. We had an interesting question from two ends of the spectrum around, for me, the probe size and also future applications of FibroScan. Where we have an active FibroScan screening program right now, and we have obviously all of the relevant probes. But one of our questions is we're regularly using the XL probe, the larger one, but we do have patients that are actually too large for that XL probe based on, I guess, our geographic prevalence of obesity in North Carolina. So my question to you is, and I think it's going to be a common question for all physicians and screening facilities and clinical trial centers, do you envision producing an XXL probe, a larger probe, or does it just not work at a certain mass?
Right. Yeah. So yeah.
Yeah.
Yeah, so this is one of the extra slides that I didn't really show, but you're right. Basically, today we have 3 probes, so an S&M and an XL, and the only difference between these probes is basically where they take their measurements relative in terms of distance from the skin to the liver, right? So as the amount of skin to capsule distance or the amount of tissue between the probe and the liver increases, you might shift to the next probe up. But obviously, especially in NASH, not so much in hepatitis C before, where our company was very involved in, we see that there are situations these days where even the XL probe won't be able to scan the patient. Although there are a lot of obesity tricks, and this is also one of the things that our clinical specialist team trains on. There are a lot of obesity tricks that you can apply to ensure that amount of tissue actually becomes less by providing the right amount of pressures, etc., and positioning. But there are internal developments right now. We call it something that it's not available for prime time yet, but we are working on something that will auto-assess the depth of measurement, and that depth of measurement will go beyond what the XL probe is capable of today. And we're anticipating that software will be available by the end of this year.
So that'll be software and not just an additional probe that measures deeper?
That's correct. Just additional software. So we're just basically making the FibroScan smarter.
Okay, and more sensitive, I guess. All right, that makes sense. And so from the XXL portion all the way down to another question on rodents. So obviously this is a clinical development capability, but the question came up, has Echosens considered the application of this technology to a non-clinical species, mostly rodents? Obviously a good rodent model would be helpful in the preclinical development of potential candidates.
Right. So I've been with the company for about 3 years now. So I don't know the exact details of that answer. I do know that we tried in the beginning a long time ago to assess fibrosis in rodents, but that we basically abandoned that project. And there might still be one FibroScan demo machine available that will be able to do so. But no, the company is not focused on that right now, given everything that's going on in the NASH space.
All right. Well, that's a fair question. It would be an interesting machine to see if it ever happened. And we have one more question here, which is the question eventually that we all are going to ask. Two questions in one, actually. What is can you comment on the inter-rater reliability of FibroScan as far as variability? And then does that actually get to the point where it may actually affect the interventional treatment of NASH? And then can you also comment on, at the same time, what you've seen as far as correlation to biopsy data?
Right. So on the inter and intra variability, there actually was a very nice paper, and I'm not showing that reference right now, but same author, basically, Fupalanchi in 2018, presented some very nice NASH CRN data on the interoperator variability and intraoperator variability of FibroScan. So I would recommend the person that asked that question to reach out to me so I can share that paper later on. But there it was clear that between the first and second examination, between different operators or the same operator, the amount of variability was actually surprisingly low. I mean, in the order of 0.2, 0.3 kPa or 0.1, 0.2 decibels per meter for CAP. So relatively low, and certainly when you look at the inter and interoperator availability of assessing a biopsy reading, I would say it's pretty on par, if not better than that. So I am presenting an older reference here with numbers around 0.7 and 0.8. So that is an older 2017 paper. So there is definitely some variability there, and what I always say is that variability goes down very quickly once an operator becomes, first of all, properly trained. That's very important that people don't train each other, that we have a dedicated team to do so, and also consistently keeps taking measurements over time. So no pause of 1 year and then start to take assessments again, because that is where the variability will slip in. It's not so much about the system, to summarize, it's more often about how the system is used where the variability comes into play.
So consistent and frequent training and retraining or use contributes to a lower inter-rater variability rating.
Exactly. Yeah, that's way more elegantly put than I did. So indeed.
And so the last question that I mentioned that we'll take right now is, can you comment on the comparison to biopsy?
Okay. So it's in comparison to biopsy, you can only do so well. So especially when you're looking at fibrosis, I'll comment on that one per se. In the advanced stage of fibrosis, our ORA curves relative to biopsy are very good. So in the 0.9 scale, when you go to F3, F2, you hit 0.8, maybe 0.75. And once you hit below fibrosis stage 2, the performance of the technology degrades. And it's not so much per se because it's FibroScan related. Any elastography technique will suffer from that, and that's just because that the relationship between the fibrosis stages and the amount of collagen content is exponential. So the difference in collagen content between F3 and F4 is huge compared to the difference in collagen content between an F1 and F2. And because we measure collagen content, obviously we have more difficulty distinguishing the earlier stage of fibrosis than the late stages.
So it's really a function of collagen sampling and its prevalence that basically increase the correlation to biopsy.
Yes, advanced fibrosis stages, a very good performance. Low fibrosis stages, a little bit less. And that is also why we are looking at score combinations where, for instance, we are able to have other parameters sort of like come into play when we're assessing the lower stages or inflammation components, etc.
Perfect. And I think that you've got quite a few questions, and obviously these questions will be readdressed in the Q&A document. The recording and your contact information will be forwarded to everyone on the call so that you can have some additional follow-up from people that have more specific questions. But I wanted to thank you for a very interesting presentation, and we'll move on to our third and final presentation. That presentation is from Clay Dean, MS. Finally, our last speaker shares the actual challenge of building databases by marrying the available technologies to screen patients with the other outreach efforts needed at the site level to identify, recruit, and retain this patient population. Clay Dean is the Vice President of Clinical Pharmacology Services at CTC with a significant background in endocrine disorders. As such, he is a member of the industry government consortium known as NIMBLE, or the Non-Invasive Biomarkers of Metabolic Liver Disease, where he shares his expertise in metabolic clinical pharmacology and operations in early clinical research. And I'm going to go ahead and turn that over to him.
Thanks, Lorraine, for the introduction. Today I've been asked to speak a bit about recruitment of people with fatty liver disease to participate in clinical trials, especially those in the early proof of concept phase of clinical development. I'll discuss some of the challenges that we faced here at our site in North Carolina, as well as some of the solutions that we found to be the most effective. To begin, it's important to understand why these challenges are presenting themselves now. NASH wasn't described until 1980. And then with the 2008 FDA-issued guidance for industry requiring thorough cardiovascular risk assessment for all anti-diabetic medications in development, innovation in that already crowded therapeutic area became increasingly stifled. Importantly, in very recent years, publication of market size estimates for fatty liver disease and the manuscript from the FDA-AASLD joint workshop, which suggested surrogate endpoints could be adequate for registration, help explain how, especially anti-diabetic drug developers, found themselves at a crossroads. That's intentionally exaggerated on this slide. It's only meant to illustrate how fatty liver disease emerged so quickly to such a large strategic business opportunity, and why so many displaced anti-diabetic drugs and developers were repurposed towards this interrelated metabolic disturbance. Ultimately, these shifts have led to a sudden and massive imbalance between the high demand and low supply of clinical trial participants with fatty liver disease. So why is recruiting people with fatty liver disease so difficult? First of all, the contract research industry has been slow to recognize and respond to shifting focus. Many of the reasons are listed here on this slide. The growth and maintenance of a database for a clinical research unit are incredibly resource intensive. Access to new patient populations tend to develop slowly and more slowly than this industry segment will tolerate. Traditional methods for database growth and expansion are not efficient enough, they're not rapid or productive enough. And in our experience, physician referrals to clinical research units are unlikely to prove adequate. And trial complexity, especially at screening, is a difficult obstacle towards patient participation. We'll discuss these points further throughout the presentation. Regarding the lack of awareness, I can remember when I first approached the senior leadership at a large CRO where I was previously employed to strategize their service offering in that space. And I was asked by the senior leadership to focus on metabolic disorders, which was my remit within the company, and not on viral ones. And it was a really eye-opening experience for me to realize how unaware people are, even those that are working in the drug development industry, about fatty liver disease. And it definitely put into perspective for me how unfair it would be to expect the general public to be very sophisticated about the condition. This slide reports the 2015 findings that fewer than 20% of people in the general population surveyed had any familiarity with fatty liver disease at all. And what I think was more surprising was the 2013 report that non-hepatologists, healthcare providers, which included internists, cardiologists, endocrinologists, thoracic surgeons, rheumatologists, and nephrologists, massively underestimated the incidence of fatty liver disease and rarely referred to hepatologists even when they did suspect fatty liver disease to be present in the patients. I think that speaks very much to why we cannot rely on referrals from healthcare providers to feed the studies at the scale that we need. So the need for a robust community outreach and education in support of clinical trial recruitment should be obvious. Often when I'm working with potential participants in studies, I find the attitude towards research as one of mistrust. They're very skeptical that they might be affected by a disease that they've never even heard of, and even if they are, they're very skeptical that it could be as serious as it is. One of the things that I found useful is to present them with a bit of a case study. In 2016, George Michael died, and the coroner listed the cause of death as dilated cardiomyopathy, myocarditis, and fatty liver disease. Whether his fatty liver disease was of non-alcoholic origins or not is debated, but personification of the condition with a prominent example has proven very useful in establishing the validity and seriousness of the condition for the participants and to get their buy-in. It really helps to facilitate that conversation. And in terms of recruitment, where are areas of potential improvement? With a condition that is this unfamiliar to the public, recruitment success is very highly dependent on consistent exposure. This, of course, includes the outreach and education that we've already discussed, but should also include exposure to advertisements for study participation opportunities. General advertising campaigns supported by multiple studies offering the opportunity to leverage efficiencies and synergy between the studies should be considered by sponsors as an opportunity, and I would hope that sponsors would become increasingly open to supporting that type of recruitment strategy at sites, especially those that are focused on serving this therapeutic indication. And then another obstacle that I mentioned earlier is the burden of the screening activity on the potential participant. Screening for studies for people with fatty liver disease can become a bit unwieldy for the participant. Cost-containing efforts are often passed on to the participants in the form of inconveniences. For example, if FibroScan or ultrasound is used as a screening gateway, much in the way that Remco described early in the screening process, as a gateway to something more conclusive, such as MRI-PDFF to confirm eligibility based on steatosis, it can add multiple screening visits to the participant's experience, and they can find it quite off-putting if it becomes very intrusive. So any opportunity that we have to reduce or compensate the participants for that burden would have a tangible impact on recruitment rates. And then finally, for multi-site studies, which many of these studies are, even in the very early phase of clinical research, sponsors may consider centralized advertising and referral campaigns to augment the site-specific recruitment activities that are going on at each individual site. We've had good luck with a couple that I'll mention, Study Kick and Subject Well, and we are also able to expand our recruitment abilities as needed with some contract recruitment service providers. And that reaches the end of my presentation, Lorraine.
Okay, thank you very much, Clay. There are a couple of questions that came across that I wanted to just walk through as well. Would you comment on the availability of biopsy-confirmed NASH subjects that originally are viewed, are requested in protocols as a first line of preference?
Sure. So in our space, we serve traditional early phase 1A, 1B. Occasionally, we have 1B proof of concept arms incorporated into a combined protocol. And then we also serve the 2A space. Occasionally, we will have a request for biopsy-proven NASH in order to determine eligibility for a 2A trial. And in my mind, I sort of separate out those opportunities into a couple of buckets. So many of the programs that we work with are focused on steatosis or reversal of steatosis as their mechanism of action, their therapeutic mechanism of action. But certainly the ones that are looking at change in fibrosis, those more often will require the biopsy. in order to determine eligibility and as an endpoint if the trial is long enough. Change to fibrosis, as Remco mentioned, does take a significant amount of time. So often in the very early phase space where we serve clinical development, the length of exposure may not be adequate for biopsy to make sense.
And so if I follow up to that question, I don't know if there's a way you could categorize or give hints to the listeners out there if they had to design a study that kind of went from easier to harder ways to qualify patients, not qualify, but create criteria to operationalize a study that ultimately wanted to show some improvement in NASH. Would you have any suggestions for them?
It's in a difficult place right now for sure, where if we are truly looking for change to fibrosis, the NIMBLE consortium that you mentioned, that's certainly what we're looking to do, is develop and validate biomarkers that can exhibit that change even over relatively short periods of time. I spoke a good bit in the presentation about the crossover between the diabetes drug development space and fatty liver disease. They are often comorbidities of each other and very connected in that regard. So I think it has been difficult for many of the drug developers that have had long careers in anti-diabetic drug development to make their peace with the fact that the endpoints associated with fatty liver disease probably will not be anywhere on the order of what they have been for diabetes, where the biomarker that we're developing drugs to and treating to is the A1C, representing a 3-month average of the glycated hemoglobin. That won't be the case with fatty liver disease. The change in fibrosis is so different, and so much longer than how responsive those diabetes biomarkers are. But I think as we continue to develop and test biomarkers that can indicate early change in fibrosis that may not be structurally detectable but are maybe detectable as a circulating biomarker, there will be potential in the industry for that, I think.
And so one final question, can you comment on the differences in the rate of recruitment based on the significance of fibrosis? So if someone's designing a protocol, does it make sense to, and they need to be able to recruit it and retain those subjects. Is there any benefit to focusing on lesser fibrosis versus more significant fibrosis, or do you think that the fibrosis is so kind of overwhelming in the disease prevalence that doesn't necessarily impact the ability to recruit those studies?
I think that will become a challenge where it may be diminishing returns to compromise on the level of fibrosis, because the non-invasive biomarkers like FibroScan are more predictive or better validated in the more advanced fibrosis than they are at the lower levels of fibrosis. So I think that at the lower levels of fibrosis, it to me seems that it may require biopsy in order to detect those changes that would occur during a therapeutic trial.
Okay.
Trial.
And so we have one additional question out there. The question actually verbatim is, how do you suggest that biopharma companies and CROs connect with NASH patients in a HIPAA-compliant manner? That's part A to the question, and that's really around outreach and education. And then part B is also, do you know if and when NASH will be a focus for the FDA PFDD meetings?
So I do not know about the focus for the PFDD meetings, but I think that there are many creative ways that sponsors can support that connection with patients with fatty liver disease that is very HIPAA compliant. Certainly, any outreach and education opportunities that they can financially support are very meaningful and would not have any implication with HIPAA. I think that the challenge there is that the return on investment may not be easily measurable or easily defined in that regard. It's almost an act of faith to move forward with it in a way that you can't directly measure. I mentioned earlier supporting general advertising campaigns at a site. That does make things a bit easier, especially in this disease space where most people don't know if they have fatty liver disease or they don't. So if you're putting ads out that are very specific to your study and drill down to your specific criteria, it probably is not helping your return very much, your recruitment rate on that. So to have more general ads that are focused on engaging the risk factors associated with fatty liver disease and having the site sort out which project they're most appropriate for, sharing the expense across multiple sponsors that way works very well. And then the other thing I would say, I think more and more, especially in this space, many drug developers are relying on small pharma and biotech for innovation. And those small virtual companies tend to be asset-centric and by default very asset protective. I've had several opportunities in relatively recent years around this drug development space to speak on local television shows or radio shows that have a health segment related to it about fatty liver disease as an education opportunity for the people that watch or listen to these radio shows or TV shows. And many of the companies that I've had the opportunity to give high-level details about their study that we were currently recruiting at the time have asked that we not do that because they are looking out for their compound. They don't want that type of visibility around it. But those types of activities are very useful for recruiting studies, and I've found that to be the case across many of the comorbidities related to diabetes, including chronic kidney disease, et cetera. If you can highlight, at least on a very high level, the recruitment the study participation opportunities in some local health segments, that's very useful.
I think that's true, but I also think that a larger kind of pan-industry effort is really needed to get to that level, because much like the NIMBLE program, where you're actually going into a positive focused consortium to understand other endpoints and other biomarkers, I also wonder if a sub-arm of a program like that couldn't be to, in a cross-industry way, start developing a resource for patients to go to actually look at clinical trials that are ongoing. Would NIMBLE or would a separate effort be an appropriate way to start building that kind of awareness for patients?
Well, yeah, I think across the board and even beyond drug development, just disease awareness in general, we've got our work cut out for us in that regard. I think if you engage the general population, you would be surprised how disbelieving they are that a disease was first defined in 1980. And, the people that are affected, usually it's within their lifetimes, and that it's reaching epidemic proportions and could have really serious health. Have a very hard time believing that. So we are definitely going to have to do a lot of work in education and awareness.
And I think there is a NASH educational foundation, but it would be interesting to link that up with pharma companies and really look at pharma biotech companies and really look at a cooperative way to educate and then give people the opportunity to opt into, all of the spaces, all of the clinical trials that are going on now. So that's just a comment, but we're really running out of time, and I think that'll be our last question. So thank you, Clay, for all, for your comments and your presentation. And in conclusion, I'd like to thank everybody for their kind attention and questions during today's webinar. And as a reminder, we will be sending out the recording and a fully responsive Q&A sheet as we promised for everyone's review on the webinar. And at that, I'd like to conclude, let you know that we will be following up with that information, and we're here for additional support in any of these areas. The information will come with contact information back to the presenters, so that you can follow up directly and also see the responses that they've prepared based on your questions. So again, we appreciate the attendance and the interaction. Thank you very much for your participation, everyone. With that, I'd like to conclude the webinar.